My name is Hope Pattison, and today we're going to be talking about epigenomics. Let's start off by breaking down the key part in the word epigenomics, genome. Your genome is your complete set of genetic information that you inherit from both parents. This genome is included in every cell that you have, and it is made of segments of DNA, called genes, that follow the genetic code. The genetic code also directs the specialization of cells. One way this happens is when certain proteins or chemical groups attach to a gene. Once attached to the gene, they chemically modify the gene's DNA. However, they don't alter the genetic code. These modifications then turn on some genes and leave other genes turned off. This process is what makes specific cells, like stomach or heart cells, in your body. Scientists track the modification of DNA for each unique cell type, which is what the cell's epigenome is. But when a cell's epigenome is disrupted, genes can be incorrectly turned on or off, which can cause diseases like cancer. This poses to be a huge problem. When cells are cancerous, they rapidly divide, and the cells being divided carry the same abnormal signals that direct the genes incorrectly. Now that we've learned about the genome and the epigenome, understanding what epigenomics is will be a little bit easier. Let's look at the word parts again. Epi means above, so epigenomics means above the genome. In other words, this includes your genetic code and the elements that define how that code is used. So, epigenomics is the study of the modifications made to the genetic material of a cell. We've already talked about what happens when a cell's epigenome is disrupted, but what does all this mean for scientists? Well, the significance in the discovery of the epigenome is that the DNA code is not the whole story for why abnormalities happen in the body. It helps explain certain phenomena, such as why identical twins can end up with different diseases, even though they have the same DNA. Because genes can be chemically altered without modifying the genetic code, this explains why one twin could get a disease and the other one doesn't. Even though they have identical genes, one twin's gene could be turned on, while the other twin's gene could be turned off. So, they have the same genome, but different epigenome. Let's go back to cancer. Cancer develops either when p53s, which are tumor suppressor genes, are abnormally turned off, or when oncogenes, which stimulate cell division, are abnormally turned on. Instead of trying to kill cancer cells completely, scientists are now attempting to change the instructions of the cell by trying to turn on the p53 gene, or turn off oncogenes. This new approach is called epigenetic therapy, and is just one example of why the discovery of the epigenome is monumental in the science world. Diseases like Alzheimer's and autism are also thought to be linked to a disruption of the epigenome, and the discovery and development of epigenomics could lead to new scientific discoveries to help cure these diseases, as well as many others. Thanks for watching.